Welcome back to our class. Last lesson, we learned about neurons and neuroglial cells. We know that our nervous system is composed of these two types of cells. These cells do not exist in isolation but are interconnected through synapses. The term synapse was proposed by the British physiologist Charles Scott Sherrington in 1897. Sherrington observed early on that if reflexes involved only continuous nerve fiber signal conduction, the reflex speed should not be as fast as observed. Limited by the objective conditions of the time, Sherrington described the synapse as a functional, not a structural, structure used only to separate two neurons. With the development of microscope technology, synapses gradually became understood. A synapse is a transcellular structure. The synapse between an efferent neuron and an effector cell is also called a junction, such as the skeletal neuromuscular junction we learned before. Based on the medium used for information transmission, synapses can be divided into two main categories: electrical synapses and chemical synapses. The human central nervous system has approximately 10 to the power of 11 neurons. Each neuron forms synapses with other neurons, ranging from hundreds to hundreds of thousands. Therefore, the total number of synapses in the CNS is astronomical. This reflects, from one perspective, the extreme complexity of communication between neurons within the CNS. Let's first look at electrical synapses. Electrical synapses use electric current as the transmission medium, and their structural basis is the gap junction. When gap junctions are open, they allow inorganic ions and many small organic molecules to diffuse down their concentration gradients from the cytoplasm of one cell to another, simultaneously forming intracellular conductive channels. In our study of cellular electrophysiology, we learned that action potentials are generated by the flow of sodium and potassium ions. Therefore, when neurons connected by gap junctions experience a local potential or action potential in one cell, ions and the potential can be transmitted to the other cell via the gap junction, causing it to undergo a potential change as well. Electrical synaptic transmission is generally characterized by bidirectionality and speed. Electrical synaptic transmission is common in the nervous systems of invertebrates. It participates in mediating signal transmission between sensory and motor neurons in escape reflexes. In the adult mammalian central nervous system and retina, electrical synapses are mainly found between cells within neuronal populations that require highly synchronized activity. Having looked at electrical synapses, let's examine our old friend, the chemical synapse. Chemical synapses use chemicals released by the neuron, i.e., neurotransmitters, as the information transmission medium, and are the most common type of synapse. They are typically formed by the axon terminal of one neuron contacting another neuron or an effector cell. Based on whether there is a tight anatomical relationship between the pre- and postsynaptic parts, chemical synapses can be classified as directed synapses or non-directed synapses. Non-directed synapses. This type of synapse lacks the structure of a classic synapse. The neurotransmitter released from the presynaptic terminal can diffuse to postsynaptic components that are relatively distant and widespread. Hence, it is also called non-synaptic chemical transmission. On the many axonal terminal branches of these nerve fibers, a varicosity containing numerous synaptic vesicles appears approximately every 5 micrometers. A single neuron can have up to 20,000 varicosities. They do not form classic synaptic connections with the postsynaptic effector cells. Instead, they reach the vicinity of the effector cells along the branches. When a nerve impulse reaches a varicosity, neurotransmitter is released from the vesicles within the varicosity and diffuses into the surrounding area. If neighboring tissue cells express the corresponding neurotransmitter receptor, their membrane potential can change under the influence of the neurotransmitter, producing what is called a junctional potential. Through this form of synaptic transmission, a few nerve fibers can innervate many neurons or effector cells. For example, postganglionic sympathetic fibers are a typical example. Non-directed synapses have a kind of broadcast nature, but our directed synapses are very specific. The neurotransmitter released from a directed synaptic terminal acts only on a very localized part of the postsynaptic membrane structure. Typical examples are the skeletal neuromuscular junction and the classic synapse between neurons. Do you remember the neuromuscular junction we discussed earlier? Yes, the neuromuscular junction consists of three parts: the presynaptic membrane, the synaptic cleft, and the postsynaptic membrane. The presynaptic membrane contains small vesicles filled with acetylcholine. The synaptic cleft contains acetylcholinesterase. The postsynaptic membrane has N2-type acetylcholine receptors. Is this also the case for directed synapses in the nervous system? The answer is yes. The structure and the process of excitation transmission are the same as what we learned before. However, the neurotransmitter contained in the vesicles of the presynaptic membrane differs among different types of neurons. There are many types of neurotransmitters in nerve cells. Over 100 neurotransmitters are known in mammals. Due to the different neurotransmitters, the corresponding hydrolytic enzymes in the cleft and the receptors on the postsynaptic membrane also change accordingly. As shown in this table, only a portion of neurotransmitters and receptors are displayed. As mentioned before, when the neurotransmitter released from the presynaptic membrane acts on the corresponding receptors on the postsynaptic membrane, it causes the postsynaptic membrane to produce either a depolarizing or hyperpolarizing postsynaptic potential, including junctional potentials, i.e., it changes the excitability of the postsynaptic neuron. When the postsynaptic membrane produces a depolarizing postsynaptic potential, it is called an excitatory postsynaptic potential, EPSP. Conversely, when the postsynaptic membrane produces a hyperpolarizing postsynaptic potential, it is called an inhibitory postsynaptic potential, IPSP. For example, during elbow flexion, the neurons controlling the flexor muscles produce excitatory potentials. This allows us to successfully perform the elbow flexion movement. Although our nerve cells are permanent cells and lack the ability to regenerate, neural synapses possess plasticity and the capacity for remodeling. From a physiological perspective, synaptic plasticity mainly refers to changes in synaptic efficacy. Additionally, it includes changes in synaptic morphology and number. Synaptic plasticity is widespread in the central nervous system. It is closely related to nervous system development and higher brain functions such as learning and memory. This includes post-tetanic potentiation, habituation, sensitization, and long-term synaptic plasticity, including long-term potentiation, LTP, and long-term depression, LTD. The regulation of synaptic plasticity provides a theoretical basis for neurological rehabilitation therapy.